Okay, so all of you, question number 151. In guinea pigs, black short hair, capital B, capital B, capital S, is dominant over white long hair, small b, small b, small s. During a dihybrid cross, the F2 generation individuals with phenotype capital B, capital B, capital S, capital B, small b, capital S, capital B, capital S, small s, and capital B, small b, capital S, small s are in a ratio of 1:2:2:4. So which are they, sir? You have capital D, capital B, capital S is 1. Capital B, capital S, small s are 2. Capital B, small b, capital S, capital B, small b, capital S are 2. And capital B, small b, capital S, small s will be 4. So 1:2:4. So this is exactly like your yellow round, green wrinkled. What is the ratio of yellow round? You have 1:2:4. Similarly, here we are having black short hair, 1:2:4. Yeah. Then 152. A tall plant was grown in a nutrition deficient soil and remained dwarf. When it is crossed with a dwarf plant, then a tall plant is grown in a nutrition deficient soil, hydroponics. अगर minerals soil कम रहेंगे soil में, the tall plant will not grow tall, it will remain dwarf. Now, when this plant is crossed with a dwarf plant, what will happen? Plant is dwarf, but golden was we have changed the phenotype by growing it in hydroponic solution, but we have not changed the genotype. So genotypically it is still capital T, and when it is crossed with a dwarf plant, small t, then capital T into small t, what will it give you in the next generation? All hybrid talls. So by changing a tall plant into dwarf, by changing the soil constitution, nutrition deficient soil, we change the phenotype, but we cannot change the genotype. So it will still remain capital T, and crossed it with a small t, the F1 generation will give you capital T, small t, all hybrid talls. Right. Then 153, the term genetics was proposed by Bateson. 154, cross between unrelated groups of organisms is called as hybridization. When two unrelated organisms are crossed, what is that called as? Hybridization. 155, a pure tall and a pure dwarf were crossed to produce offspring. Offsprings were self-crossed. Then find the ratio between true breeding tall and true breeding dwarf. Pure tall and pure dwarf are crossed, and what will we get in the F1 generation? Capital T, small t. And F2 generation में क्या मिलेगा? You will get capital T, small t, and here also you will get capital T, small t. This will become capital T, this will become capital T, small t, this will become capital T, small t, and this will become small t, small t. तो ये दोनों कौन है? Capital T, small t, hybrid. और ये दोनों कौन है? Tall and dwarf, pure breed. And what is the ratio of pure breeding tall, capital T, 1, and pure dwarf, small t, 1? So what is the ratio we will get? 1:1. So what ratio we get? 1:1. Then 156, on selfing capital R, capital T, small t, we produce 400 plants. Find out the number of plants with genotype capital R, capital T, small t. How many plants with capital R, capital T, small t we get? 4. Capital Y, capital R, small r. So capital Y, capital R, capital Y, capital R, small r. So उसकी जगह instead of capital Y and R, we are using capital R, capital T, small t. How many we will get? 4. So 4 out of 16 are are dihybrid. Both are hybrid. 4 out of 16 are dihybrid, so that is 1 out of 4. So out of 400, how many we will get? 100. Out of 400, how many we will get? 100. So on self-link capital R, small r, capital T, small t, we produce 400 plants. How many are capital R, small r, capital T, small t? 100. 157. Genotypic ratio of a monohybrid cross is 1:2:1. 1:2:1. 158. In a dihybrid cross between capital R, capital R, capital Y, and small r, small y, small y, number of capital R, small y, small y in the F2 generation will be 4. 4 of them are dihybrid. 159. In a pea plant, yellow seeds are dominant to green. If a heterozygous yellow seeded plant is crossed with a green seeded plant, what ratio of yellow and green plants would you expect in the F1 generation? 50:50. Capital Y small y into small y. So it's an example of a monohybrid test cross. And what monohybrid test cross ratio we get? 1:1, that is 50:50. 160. Two genes, capital R and capital Y, are located very close on the chromosomal linkage map of maize plant. When capital R, capital Y, and small r, small y genotypes are hybridized, then F2 generation will show a higher number of parental types. Because the capital Y, capital R are located close on the same chromosome, then what will happen in the F2 generation? When it will undergo formation of gametes, capital Y and capital R are linked. So linked genes will not show independent assortment. So you will not get recombination, you will get parental combination. So what will capital Y, capital R on the same chromosome located tightly to each other show? Parental combination, will not show recombination. So higher number of parental types. 161. A common test to find out genotype of a hybrid is crossing of the F2 progeny with the male parent. So do we remember हमने जब क्रॉस किया था टॉल प्लांट को इमैक्युलेट करके we made female और dwarf plant को we made male. So what is dominant plant usually? Female. What is recessive plant usually? Male. तो टेस्ट क्रॉस में हम लोग क्या करेंगे? We will cross the F1 generation or F2 generation with the recessive plant. In other words, which plant? Male plant. So we will cross it with recessive plant. In other words, the male plant. Then 162. Which one of the following characters studied by Mendel and Greenfield was found to be dominant? Inflated pods were dominant, all the others are recessive. Inflated pods are dominant. 163. In the given plant, red color capital R of fruit is dominant over white small r, and tallness capital T is dominant over dwarfness small t. If a plant with genotype capital R, capital T, small t is crossed with a plant of genotype small r, small t, what will be the percentage of tall plants with red fruits in the next generation? So we do the cross, we have capital R, capital T, small t into small r, small t. तो इसमें से gametes कौन से बनेंगे सर? इसमें से बनेगा capital R, capital T and capital R, small t. और इसमें से gametes कौन से बनेंगे सर? You will have small r, small t. And third, so what will we get in the F2 generation? In the F2 generation, we will get small r, small t. So you will get capital R, capital T, small t, capital R, capital T, small t. And here we will have capital R, small t, capital R, small t. So sir, what percentage will be tall plants with red fruit? Tall plants with red fruit, 2 out of 4. 2 out of 4, in other words, 50% will be tall plants with red fruit. 164. 1:2:1 phenotypic and genotypic ratio is found in blending inheritance, which is also called as incomplete dominance. Red and white से pink बनता है, that is called blending inheritance or incomplete dominance. 165, genes when present in homozygous condition results in non-viable progeny. The factor responsible for such conditions are called as lethal genes. If you remember, HbS with another HbS in a homozygous condition produces non-viable progeny, death of the child. Now what is HbS gene called as a lethal gene? So any lethal gene in a homozygous condition produces death of the progeny. What are such genes called as lethal genes? 166. Sometimes there are more than 2 alleles for a given chromosome locus. In that case, the trait is controlled by multiple alleles. 2 alleles से ज्यादा है, I capital A, I capital B, small I. तो 3 alleles 2 locus पे, 3 alleles 2 locus पे है. What is that an example of? Multiple alleles. And then 167, red flowered heterozygous is crossed with a white flower. Then what will happen if a heterozygous red is crossed with a white flower? It's an example of a test cross. So you will get 350 red and 350 white. It's an example of a test cross. 168, when a cross is conducted between black feathered hen and white feathered cock, blue feathered fowls are formed. When these fowls are allowed for interbreeding in the F2 generation, there are 20 blue fowls. What would be the number of black and white fowls? Mark this important. Black and white से बनता है blue. This is an example of incomplete dominance. Black and white से blue बनता है is incomplete dominance, and blue when it is allowed to interbreed, what will be the F2 ratio we will get? 1:2:1. What phenotypic ratio for incomplete dominance? 1:2:1. So अगर 1 black है, 2 blue है, 1 white है. So if there are 10 black, there will be 20 blue and there will be 10 white. So if blue are 20, so black and white कितने होंगे? 10 and 10. So 10 black and 10 white. Nice question. Example of incomplete dominance. 169, alleles are alternative forms of a gene. Alternative forms of a gene are called as alleles. 170, very nice question. One of the parents has a mutation in a mitochondria. In that cross, if the parent is taken as a male, during segregation of the F2 progeny, the mutation will be found in none of the progenies. So, mitochondria में खुद का DNA होता है, और अगर उसमें mutation हो गया, then what will happen? If a mutation takes place in a mitochondria, समझो the mutation takes place in a male mitochondria, then will the mutated gene pass in the next generation? I hope you remember, when a sperm enters the egg, then only the nucleus of the sperm enters the egg. The mitochondria falls off along with the middle piece and the tail. So male mitochondria का inheritance female के अंदर, egg के अंदर नहीं होगा. So none of the progenies will show the paternal or the male mitochondria characteristic. Rather, the egg will always inherit maternal mitochondria. And that maternal mitochondria, egg का mitochondria is the mitochondria of zygote. Sperm का only nucleus goes in, egg का mitochondria zygote से inherit होगा. So maternal inheritance of mitochondria is always there. So paternal inheritance of mitochondria is never there, and maternal inheritance of mitochondria is always there. And this phenomenon, please write it in your book, is called as cytoplasmic inheritance. इसे क्या कहते हैं? cytoplasmic inheritance is what this is called as. And समझो the same question was asked. One of the female parents has a mutation in the mitochondria. If that cross parent is a female, then during segregation of F2 progeny, the mutation will be found in, what will be your answer? All the progeny. If the mutation of mitochondria is in male, then it is found in no progeny. Because the mitochondria of the sperm falls off with the middle piece and tail, तो वो male parent से mitochondria किसी भी बच्चे में नहीं जाएगा. And egg के mitochondria में mutation हुआ, then it will be inherited in the zygote of all the children. And what is that phenomenon called as? Cytoplasmic inheritance. 171. A test cross involves crossing of the F1 hybrid with the double recessive genotype, and that is called as a dihybrid test cross. So see, crossing of F1 hybrid with double recessive genotype is a dihybrid test cross. 172. Which of the following traits of garden pea studied by Mendel was a recessive feature? Green seed color is recessive, yellow is dominant, बाकी green pod color, round seed color, axial flower position are all dominant. Only green seed color is recessive. 173. A plant of F1 generation has genotype A capital, B capital, B small, C capital, C capital. On selfing of this plant, the phenotypic ratio in the F2 generation will be, I hope you remember, last lecture, first thing I had said, types of phenotypes is 2 raised to n, where n is heterozygous. Types of phenotype 2 raised to n, where n is heterozygous. तो यहाँ पर n कितना है? A is homozygous, C is homozygous, only B is heterozygous. Only B is capital B, small b, heterozygous. So how many types of phenotypes are there? 2 raised to 1. 2 raised to 1. So how many phenotypes? Only 2 phenotypes. Only 2 phenotypes. And what will be the phenotypic ratio in the F2 generation? It is the monohybrid cross. It is not a dihybrid or a trihybrid cross. And what will be the phenotypic ratio in the next generation? 3:1. So if it's a monohybrid cross, only B is hybrid. Only B is hybrid. So it is monohybrid cross. So what will be the phenotypic ratio in the F2 generation? 3:1. 174. Inheritance of ABO blood group shows multiple alleles. ABO blood group is multiple alleles. 175. When different alleles of a gene are present in an individual, the individual is heterozygous. If it is different allele, capital T, small t, दोनों present है, then what is a different allele, capital T, small t, called as heterozygous. The organism chosen by Mendel to explain laws of inheritance is Pisum sativum, the garden pea plant. 177. In a monohybrid cross involving incomplete dominance, the phenotypic ratio equals the genotypic ratio in the F2 generation. The ratio is 1:2:1. In incomplete dominance, was the ratio 1:2:1. 178. How many different kinds of gametes will be produced by a plant having genotype A capital, B capital, B small, C capital, C small? If you remember, types of gametes is 2:n, where n is heterozygous. So यहाँ 2:2, क्योंकि B is also heterozygous, C is also heterozygous. So 2:2, how many gametes will be produced? 4. How many gametes are produced? 4. 179. In Mendel's experiment with garden pea, round seed shape capital R was dominant over wrinkled seed small r. Yellow cotyledon capital Y was dominant over green cotyledon small y. What are the expected phenotype in the F2 generation of a cross capital R, capital Y into small r, small y? You will get 9 round seed with yellow cotyledon, 3 wrinkled seed with yellow cotyledon, 3 green seed with yellow cotyledon, 3 green with round seed with green cotyledon, and 3 wrinkled seed with green cotyledon. So 9 round and yellow, 3 round and green, 3 yellow and wrinkled, and 1 green and wrinkled. So what will be the answer for this one? What is the type of phenotype in the F2 generation? Answer D, round seeds with yellow cotyledons and wrinkled seed with yellow cotyledons, and as a matter of fact, round seed with green cotyledons and wrinkled seed with green cotyledons. All 4 in the ratio of 9:3:3:1. 180, phenotype of an organism is the result of answer genotype and environmental interaction. Capital T से क्या बनेगा? Tall. So genotype capital T will make it tall. And environmental interaction. If you grow capital T in a nutrition deficient soil, what will it become? Dwarf. So environmental interaction से वो dwarf बन जाएगा. So capital T makes it tall, but soil without nutrients it will become dwarf. So genetics and environmental interactions affect the phenotype. What is incorrect for Mendel's realm? Discovered linkage. No, Mendel did not discover linkage. He was lucky that there were no linked genes, otherwise he did not know about linkage. 182, jumping genes in males were discovered by Barbara McClintock. In maize plant, she found genes which jump from one chromosome to other, and such jumping genes are called as transposons. Transposons कहते हैं, which are called as jumping genes, and Barbara McClintock found jumping genes in maize plant, and they were also called as transposons. Then Nielsen proved polygenic inheritance in wheat kernel. Polygenic inheritance. 184, when a tall and dwarf plant are crossed, from which cross 1:1 ratio is obtained? Answer A, capital T small t and small t is an example of a test cross. 185, in Mirabilis, a hybrid for red, capital R, and white, small r, flower produces pink, capital R, small r flower. A plant with pink flowers is crossed with white flowers. What is the expected phenotypic ratio? So pink, capital R, small r into white, small r. What is this an example of a test cross? And what is the ratio you will get? 1:1. So what will be 50% pink and 50% will be white. 50% pink and 50% white. 186. Ratio of progeny when a red color heterozygote is crossed with a white colored plant in which red color is dominant to white color will be 1:1. Red color heterozygote means is capital R, small r, crossed with a white flower, small r. So what is this also an example of a test cross, and what ratio we will get? 1:1. 187. Leaf color in Mirabilis jalafa is an example of a non-Mendelian inheritance. लीफ कलर का मेंडल का कोई contrasting characteristic नहीं देखा था मेंडल ने, so leaf color is a non-Mendelian inheritance. 188. The F2 generation offspring in a plant shows incomplete dominance exhibit a similar phenotypic and genotypic ratio of 1:2:1. 189. 2 pea plants were subjected for cross-pollination. Out of 183 plants produced in the next generation, 94 plants are tall and 89 plants are dwarf. The genotypes of the 2 parental plants are likely to be 94 tall, 89 dwarf means approximate ratio is 1:1. Agar approximate ratio of tall and dwarf is 1:1, what is this an example of a test cross? So what will be your answer? Capital T small t into small t small t. Capital T small t into small t small t. So that's C. 190. Mendel observed that a certain character did not assort independently. Later scientists found this is due to linkage in traits. अगर 2 जीन्स एक ही क्रोमोसोम पे लिंक्ड है तो उनका इंडिपेंडेंट असॉर्टमेंट नहीं होगा. Mendel did not know that. Other scientists found that is called as linkage. Then 191. How many types of gametes are produced by genotype? Capital T small d, capital E small e, capital F small f. Types of gametes 2 raised to n. और यहाँ पर n heterozygous कितने हैं? D is heterozygous, E is heterozygous, F is heterozygous. So 2 raised to 3. So 2 raised to 3, how many gametes will be produced? 8. 8 gametes will be produced from this genotype. 192. What types of gametes will be produced by capital R, small r, capital Y, small y? Capital R, small r, capital Y, small y will produce capital R, will produce capital R, capital Y, capital R, small y, capital R, small y, small y. So what will be produced from capital Y, capital R, small r? The 4 types of gametes. 193. Select the statement which is not correct. We go in the reverse order. ABO blood group is an example of multiple allelism. That is true. Height, weight, intelligence, skin color are polygenic inheritance. That is true. In case of polygenic inheritance, many intermediate phenotypes are found between two extremes. Skin color we had done, black, white के बीच में बहुत सारे colors थे. Dark brown, fair, very fair, etc. So that is true. Polygenic character is controlled by a pair of alleles. No, polygenic character is not controlled by one pair of alleles, but by 2 pairs of alleles or by 3 pairs of alleles. That is called as polygenic inheritance. So it's not a pair of alleles, it is 2 or 3 pairs of alleles. 194. Mendelian recombinations are due to independent assortment. Why will we get recombination? Because of independent assortment. 195. Pure tall plants are crossed with pure dwarf plants. In the F1 generation, all the plants were tall. These tall plants of F1 generation are self, and the ratio of tall to dwarf obtained was 3:1. This is because of law of dominance. F1 में सब tall हो गए और F2 generation में 3 tall 1 dwarf आएगा. That is because of law of dominance. 196. Which of the following animals is most commonly used in experiments in genetics? Answer: fruit fly. Fruit fly or Drosophila is the most commonly used experimental material. So nowadays जितने भी genetic experiment करते हैं, what do we do? Use a fruit fly or Drosophila. 197. Genes are present on chromosomes. Genes are found on chromosomes naturally. 198. Which of the following law was discovered by Mendel first? Answer: law of segregation. Mendel ने पहले law of segregation discover किया था. Then why do we call it second law? Because law of dominance is expressed in F1 generation, law of segregation is seen in F2 generation. So law of dominance is called first law and law of segregation is called as second law. But historically, which was the law Mendel first discovered? Law of segregation Mendel historically first discovered. 199. In Drosophila, gene for white eye mutation is also responsible for depigmentation of body parts. Such a gene that controls several phenotypes is called as a pleiotropic gene. So gene for white eye mutation, there is a gene for white eye mutation, तो उसमें eyes white हो जाएगी और body पर depigmentize हो जाएगा. तो एक ही gene से eye भी white होती है, body भी white होता है. What is such a gene called as a pleiotropic gene? One gene controlling multiple phenotypes is a pleiotropic gene. 200. Very nice question. The total number of progeny obtained through dihybrid cross of Mendel is 1280 in the F2 generation. How many are recombinants? Now it is given 9x is to 3x is to 3x is to x is 1280. So this is 1280. In other words, 16x is 1280. So x will become 80. X will become 80, and the question is given how many are recombinant. 3x and 3x are recombinant. 9x and x are parental. 3x and 3x are recombinant. So 6x is 80 into 6, that is a total of 480 are recombinant. 480 will be recombinant. And 201, the ratio of 1:6:15 is to 20:15:6:1 is associated with, your answer is polygenic inheritance. What is that called as? Polygenic inheritance. And with that we have finished off here, but all of you, I would want you all to write down certain other ratios. So certain other ratios I want you all to write down, और ये प्लीज ध्यान से आपके बुक्स में लिख लेना. Write it down carefully. You have a phenomenon called supplementary genes, complementary genes जो हमने सीखा था. साथ में we have a phenomenon called as dominant epistasis. Dominant epistasis ये आपके NCERT बुक्स में या नोट्स में दिया नहीं है. Just take a few examples of it. Fruit color in cucurbita pepo. Fruit color in cucurbita pepo, and also found in coat color in dog. Coat color in dog. Write this down, all of you, in your notes. Fruit color in cucurbita pepo, coat color in dog, and this ratio is 12:3:1. So what is the ratio for dominant epistasis? 12:3:1. And then we have something called as recessive epistasis. Recessive epistasis is found in coat color in mice. Coat color in mice and pigmentation in onion bulb. Pigmentation in onion bulb. And this ratio is 9:3:4. 9:3:4. And something called as duplicate genes. Duplicate genes which is found in fruit shape in shepherd's purse. Fruit shape in shepherd's purse, which is also called as Capsella bursa. Capsella bursa, and the ratio is 15:1. Ratio is 15:1. इसके डिटेल्स आपके NCERT में दिया नहीं है, नोट्स में दिया नहीं है, तो we won't go with the details. Just extra information. Suppose they ask you what is the phenotypic ratio of dominant epistasis? Answer is 12:3:1. Which plants show it? Fruit color in cucurbita pepo and coat color in dog will show dominant epistasis. Recessive epistasis का phenotypic ratio क्या है? 9:3:4. Where do we find it? Coat color in mice and pigmentation in onion bulb. They will show recessive epistasis 9:3:4 ratio. And duplicate gene, the ratio is 15:1. And where do we get it? Fruit shape in shepherd's purse, which is called as Capsella bursa. ये नोट डाउन करके लिख देना है. Just in case hypothetically they ask you in the NEET exam, you should know what are the ratios. 12:3:1 for dominant epistasis, 9:3:4 for recessive epistasis, 15:1 for duplicate gene. So with that we have finished off with Mendel's the entire chapter with all the back MCQs. Next session we will start with a very nice chapter, fun chapter, chromosomal basis of inheritance. Please study this well. Very soon a test on the entire chapter of genetic basis of inheritance, that is Mendelism. We will have the entire chapter covered up. Take care. God bless.